This is a detailed protocol of culturing periodontal ligament cell spheroids by Chitosan films. Compared with a conventional tissue culture system, the spheroid culture system produces periodontal ligament cells with increased self-renewal and orthogenic differentiation abilities.
To begin, prepare 1% weight-to-volume Chitosan solution as directed in the manuscript. Add 0.5 milliliters of Chitosan solution into each well of a 24-well tissue culture polystyrene plate. Dry the plate in an oven at 60 degrees Celsius for 24 hours to form Chitosan films. To neutralize the Chitosan films, add 0.5 milliliters of 0.5 normal sodium hydroxide to each well of the 24-well plate, and incubate at room temperature for 2 hours. Then, wash the Chitosan films 3 times with double distilled water. Sterilize the Chitosan-coated 24-well plate in 70% alcohol overnight at room temperature. Rinse the plate 3 times with PBS, then leave it under ultraviolet light overnight. Prior to cell seeding, pre-warm proliferation medium, PBS solution, and trypsin EDTA solution to 37 degrees Celsius. Discard the supernatant from the cell culture flask, and wash the confluent periodontal ligament, or PDL, cells with 10 milliliters of PBS. Discard the PBS and add 1 milliliter